Derek and Piyush, your Mobius model is relevant, needed, and helpful. It'll not surprise you to know that I have a range of questions for you.
Next comes the Mobius model, and this is the really fun part. So in the Mobius model, we've got two key components. The first is pre-training, and the second is the mixture of experts part.
There's just a lot that epigenetics can tell you about your current health and likely path forward.
Welcome back to the Human Life Expectancy Channel. I'm your host, Tom Nodine, CEO of Human Life Expectancy, Inc. Today we're talking with Derek Jacoby, who does research at the University of Victoria and who also serves as our Chief Digital Officer at Human Life Expectancy, Inc. Derek and his associate, Piyush Acharya, just published a study in IEEE called Mobius, Mixture of Experts Transformer Model in Epigenetics of ME/CFS and Long COVID, which distinguishes between ME/CFS, Long COVID, and control groups with over 97% accuracy. I'm delighted to have them join us today to discuss this study and its important implications for healthcare and for identifying novel biomarkers, which is core to what we do here at Human Life Expectancy, Inc. Welcome, Derek and Piyush.
Thanks, Tom.
Thanks for having us, Tom.
Just to bring our listeners up to speed, myalgic encephalomyelitis, or chronic fatigue syndrome, or ME/CFS as it's sometimes called, and long COVID are chronic, debilitating post-infection diseases that affect up to 470 million people. These diseases can be tough to tell apart. There are currently no validated blood or imaging tests for their clinical diagnosis. As a result, currently approximately 90% of ME/CFS patients are incorrectly diagnosed as long COVID patients, resulting in delayed care and millions of dollars in healthcare burden. So, Derek and Piyush, your Mobius model is relevant, needed, and helpful. It'll not surprise you to know that I have a range of questions for you. Do you mind if we just jump in?
Jump on in.
Great. So, how did you decide to do a study in this area?
Yeah. Let me give you a quick background that we were looking at these two conditions, long COVID and ME/CFS, and realizing that from a symptomatic perspective, it was incredibly hard to tell them apart. Each of them are made up of a cluster of different symptoms that are really very similar in both conditions. And we were looking for something that might be separated epigenetically in a way that it certainly wasn't able to be from a symptomatic perspective. But as we started to look at the literature, realized that the classical statistical techniques for doing so weren't really good enough, that they were getting about 3/4 of the cases separated properly, but the remainder were really falling out of the mix, even in the best studies that we could find out there. And we thought we could do better based on some of the deep learning research and transformer-based techniques that we've been using in other domains. And so that's what Mobius is a transformer-based classifier that is able to distinguish between ME/CFS patients, long COVID patients, and people who have neither illness.
Mm-hmm. And can I ask you, what made you think that an epigenetic test might be able to tease out the difference between these two diseases?
Well, in part, the work that you and I have been doing at HLE, I have an epigenetic lens on over a lot of things in the world these days. But what was really interesting looking at the literature is that the underlying mechanism of ME/CFS and long COVID appears to have some differences. And there's been a lot of research on both of these conditions, even though there isn't a good diagnostic to pull them apart in the clinic. And it seems like that research had pointed to different pieces of different pathways being involved. And that really leads to the suspicion that epigenetics is a good lens at which to look at these, because anytime there is differential pathway involvement, there's probably differential epigenetic involvement.
Great, thank you. Where did the name Mobius come from?
That was Piyush's contribution, actually. Came up through thinking about the Mobius strip, which has two sides that are connected through a twist in that strip, that the symptoms are so closely related in long COVID and ME/CFS that they really do seem like two sides of the same illness. But underneath, they're really not.
That's great. Piyush, can you tell us where the data came from and how you processed it?
Yeah, that's a great question, Tom. So our study used 14 distinct data sets from the NIH's NCBI Gene Expression Omnibus. And what we've done is we've decided to look at DNA methylation data, which, as you know, Tom, is a form of epigenetic data. Now, it's where methyl groups attach to DNA molecules, and that's what we call DNA methylation. Now, we used 852 total samples, which means that there's 284 ME/CFS samples, 284 long COVID samples, and 284 healthy control samples. But there are two key things I want to note regarding the data here. The first is that all ME/CFS data comes from pre-2019. And this means that, and this is crucial because this prevents those ME/CFS patients from also having long COVID. So that's why we decided to take all of our ME/CFS samples from before 2019. And the second thing I want to note is that all of our data is publicly available from the NIH. And this means that it's prone to batch effects. Like each lab, has different protocols for conducting their experiments, and all of them use different experiments on different machines. And this leaves a lot of variation in the data. And that's why we had to use a very rigorous pre-processing technique that we've outlined in our paper.
Thank you very much. Given that data, how does the model actually work?
Yeah, so we've got our data, right? We've got 852 total samples. Then we move on to the pre-processing step, and that's where we rigorously, normalize all the data, ensure that everything is standardized across all samples, all machines. And I forgot to mention this earlier in the data collection step, but all the data is collected from the widely available Illumina 450K array and the Epic 850K array. So this is very standardized and available worldwide in a lot of labs. So once that's done, and we pre-process the data, next comes the Mobius model, and this is the really fun part. So in the Mobius model, we've got two key components. The first is pre-training, and the second is the mixture of experts part. So pre-training, I'm going to explain it in a slightly technical way, and then we'll use an analogy to better understand it for your audience. So pre-training is where we, give the model all the data, and we just throw it at it, and we don't tell it what the problem is. For context, the problem is, differentiating ME/CFS and long COVID. We don't tell it any of that. We take all the data and we give it to the model, and we tell it to learn about the data and understand the data and the relationships between different samples. And so that's the pre-training step. Next comes the mixture of experts layer. And here's where I want to use an analogy. So in the US Supreme Court, right, that's the highest level of our court system. And so what happens is that lower courts first decide which cases get seen by the Supreme Court, right? They look over all the cases, they do their thing, and then they send it up to the specialists. So when lower courts look over the cases, that's an analogy for pre-training, right? Where they sift through all the paperwork, they look through all the cases. That's pre-training, the first step. The second step is the mixture of experts part, and that's where the Supreme Court justices look over the cases, and the judge with the best background and the most well-formed opinion will contribute to the final result in the highest, most impactful way. And that's how the mixture of experts part works.
So Piyush, how do you actually train a transformer?
Yeah, so the core idea here is that we are using structured data. So we've got long COVID samples labeled, we've got ME/CFS samples labeled, and we've got healthy controls labeled. And we feed all of those labeled data points to the transformer model. And a good visual of this is figure 1. What we see here is a good separation of the data, and it's color-coded. And so the task of the transformer is to figure out what differences in the data enable those differences to show up there. And so it's trying to separate the three conditions using statistical techniques.
One thing that actually I'll throw in on that is that figure 1, where we're showing the separation, is based on a technique called TS&E, which interestingly came out of Geoffrey Hinton's lab at the University of Toronto, who's widely known as one of the founders of the deep learning revolution. So it is actually already a fairly advanced technique for doing a separation of this data, but it all comes from prior to the transformer revolution in deep learning. And so the engine that we're using is taking the statistical separation power 1 degree further than even the TSNE solution that we're seeing in Figure 1.
Great. Thanks to both of you. How do you test the training to make sure you've done a good job?
Yeah. So that's a good question. So for context, current doctors in a clinical setting get an accuracy of around 58%. And that's pretty low. And that's a big problem, as you were talking about.
That's almost a coin toss.
Yeah, that's right. And but that's not a fair comparison for comparing our statistical computer, right? Our AI. And so what we should do is compare the model to other types of models. We used, again, the transformer architecture, and that's a model that requires a ton of compute and power in order to train. And whenever in computer science you have to use that amount of power and that amount of complexity, you need to have some sort of justification for doing so. And that's why we first tested with simple models like logistic regression, which got accuracy in the upper 70s. And we also tested XGBoost and random forest, which got accuracies around 80% and slightly above. But that's still not good enough for deployment in diagnostic tools and research. So that's why we developed our transformer with the mixture of experts, pre-training, and adaptive computation time. And that's what enabled our model to receive an accuracy of just above 97% on an external holdout dataset.
That's amazing. But can't you get misleadingly high explanatory power by overfitting the data? How did you ensure that didn't happen here?
Yeah, and that's a great question, Tom. It's very important to prevent overfitting because for your audience who might not know what overfitting is first, let me explain it briefly.
Go ahead, please.
Yeah, so overfitting is when a model tries to memorize the data in a way. Think of taking a test, right? If you take a test and if you're preparing for a test, right, you might take practice tests. When you take that practice test, you don't want to memorize the answers to the practice test. That would be horrible because on the actual test, you would do horrible, right? But if you understand the ideas in the practice test, then you'll perform well on the real test. And so overfitting is the first example that I mentioned there. We want to avoid that at any cost. So there's a couple of things we did. So the first is that we took all of the data from all of the data sets and we combined it into one big giant data repository. Then we held out 20% of the data for testing, and 80% of the data was used for training. Now, that 20% was never touched at all during that training process. It's like hiding the answers to the real test from the model. It's not allowed to see it. And so once we trained the model, we then gave it the real test, and that's where the 97% accuracy came from. Another thing is we use tenfold cross-validation, which is a technique in machine learning where you kind of shuffle the data, which produces the impact of individual data points on the final result. So all in all, overfitting is not a concern here. And the graphs prove that, and we've taken a lot of measures to prevent that.
That's great. Are there other ways you know that your model is better than others?
Yeah, let me take that. One of the things that I think is kind of neat is that looking at the classical statistical tests that we include here, logistic regression, random forest, and XGBoost. Logistic regression is a fairly simple test in a lot of ways. You're simply looking at the linear patterns to suggest an outcome. It's fairly good with explainability, that if you have one element of your input data, you're fairly easily able to trace what impact that had on your output data. But it's also fairly simple, and so it lacks the statistical power of some of the more advanced techniques. When you go into random forests, what you're basically doing is creating decision trees out of the paths that your logistic regression is following. And those decision trees essentially walk down the variables that are most diagnostic for making your decision. So a random forest model actually maintains a lot of the interpretability of the original logistic regression models. This is not true when you come into the XGBoost model, which can be thought of from just a conceptual level as a field of decision trees. Since you have this more complex organization, you've got more statistical resolving power. In our data, about 2% more than a random forest. But you don't have the explanatory power. The data is too confusing in that presentation to really allow you to have a good explanation of what was going on in the model. One of the really neat things about the multi-headed attention model that is our mixture of experts model here is that you're able to observe inside the attention heads to identify those epigenetic CPG islands or epigenetic sites that were most diagnostic in making a decision for these different classes. And what that lets us do is reestablish.
So it's much less of a black box.
That's right. We reestablish the interpretability of the model with the transformer that we had lost through some techniques like XGBoost. And that's actually the focus of our current work, is trying to use that hinting from the attention heads as an indication of what pathways to look for downstream activity on to validate what the underlying cause of long COVID and ME/CFS are and how they differ from each other.
Yeah, for listeners that may not know, in the past, it's been difficult to explain what's actually driving the results of epigenetic tests. These tests are sometimes dependent on hundreds or even thousands of CPGs, which can frustrate those who are looking for a single answer as to why a result was achieved. So, as you're saying, Derek, linking the results to specific scientific factors is important.
Yeah, I do like that explanation, but there's actually a subtlety here that I want to elaborate on just a little bit, that there are some approaches in epigenetics that are looking for the biomarker, looking for the epigenetic site that is diagnostic for a condition. And we very much are not doing that in this model. The reason that in figure 1, you see some of the elements overlapping with each other is that there's not a clean separation in a given site. It's not like we can say this site is fully predictive of this condition. That in fact there's an intermingling of factors, and we're able to say that the primary factor in ME/CFS may be different than in long COVID, but there's a whole chain of additional sites that are being used diagnostically, some of which do have overlap between the conditions, and that means underlying disease progress, disease causes are in fact intermingled to some extent. And this makes what we're doing from a statistical separation very different than a single biomarker presentation that some labs have tried to do.
Thanks for making that distinction. That makes sense. So, at the end of the day, how much better is the Mobius model? You had mentioned 97.1%.
Yeah, so that 97.1% that we're claiming is our end classification difference is good enough to start to use clinically. And we can't say that that's ready to use clinically. This is an early statistical paper. This is looking at methods. There are a lot of steps to go through before this is ready for the clinic. It has to be validated in the real world. But I think that it is important to start it on its path. One of the things, particularly in the long COVID literature, that I've been very impressed by in recent years is the amount of work at alleviating long COVID symptoms. There have been, for instance, papers on the microbiome impacts on long COVID and using probiotic mixtures that are incredibly effective at relieving some of the long COVID symptoms. There was a study out of Hong Kong University about a year ago that showed about a 2.2 odds ratio over a 6-month period for people treated with one of these probiotic mixtures that spiked the bifidobacterium production in their patients' microbiome. There are equally some early suggestions at treatments for ME/CFS, but they're different. And because there's a different underlying mechanism, and there are things you can do for each of the conditions, in order to have people have effective symptomatic relief, we need to be able to separate the underlying disease conditions. And I think that this starts to get to the level of statistical power that we're ready to begin doing that.
That's great. And so ultimately the hope is that we'll be able to develop a new diagnostic test for 470 million people. You mentioned, though, that we're a ways away from that.
Yeah, that's right. How far we are away from that, in some sense, depends on the regulatory regime under which you operate. Because we are using a lab test that is widely available, you can go into your LabQuest or Doctor's Direct or wherever you are in the country you are, and say, "I'd like a epigenome array," and can pay your couple $100 for it, and have your results on your memory stick. And what do you do with those? As an individual, you can talk about them with your doctor, but your doctor is not going to really know what to do with them. You could run them against our classifier. This is an open mechanism. We've open sourced the techniques to do this, but you can't really have your doctor diagnostically do anything with that. Of course, a lot of the treatments for these conditions are really home remedy level treatments. The bifidobacterium yogurt is something that I make in my basement and have with my breakfast cereal. It's not a drug.
Derek, knowing you as I do, I am not surprised that you do that. I'm not at all surprised.
So there are things that you can do as an individual that don't necessarily fall under a medical regulatory regime. But it is really important to note that none of this is real medical advice until it goes through a medical regulatory process.
Absolutely.
And that's something that is just going to take longer. And quite honestly, that as a pair of academic researchers, Piyush and I aren't necessarily the ones to take it through the full course of action. So we're looking for partners, organizations that might be in a position to do some of the modern data trials where we actually have the harder task of separating long COVID from ME/CFS, possibly from SARS-CoV-2 triggered ME/CFS, in that ME/CFS itself is a post-viral condition, and SARS-CoV-2 can certainly trigger that condition. There are different conditions that...
Can I interrupt you?
Please, yeah.
With your permission, can we put some contact information in the notes section of this video so that anyone interested in exploring partnership can reach you?
Oh, absolutely. Yeah, no, I look forward to contact information being there. I also have a nonprofit organization that is involved in some of this research in British Columbia, and we do a little bit of a podcast series on that nonprofit. And so I would actually love to plug my podcast during this and throw that in there as well. We may have some follow-ups that might be interesting to your audience.
Let's definitely do that. So let me ask you a broader question. Can Mobius be used for other things?
Yeah, I'm actually really excited about the underlying technique that we have here. And Piyush explained a little bit about the way of training Mobius, how basically we took the labeled data sets and took a holdout data set that we used to later test it and trained the engine on those labeled data sets. That means that basically anything that we can collect labeled data for, we can use this as an approach to exceed classical statistical techniques in performing a separation across that data. And there are a lot of confusable things in the world, particularly when we talk about epigenetics that have so many factors involved. You might want to know if you have quit smoking long enough to be classed like a non-smoker in terms of your risk profiles. Epigenetically, we can probably see that if we collect the right sets of data and start to stratify those risk profiles. There's just a lot that epigenetics can tell you about your current health and likely path forward that we haven't previously been able to identify separations between.
Yeah. And can it also be used, as you were talking about before, to identify underlying biological pathways?
I can't make as strong a claims on that one yet. We have really tantalizing evidence that it is going to be able to, because we can look inside our attention heads and see which CPG islands are most diagnostic, most leading to the decision. At an epigenetic level, we can say that we have good hinting towards a pathway. But before you can really say that the pathway is a cause of an illness, you can't stop at epigenetics. You really have to go through to transcriptomics. You may have to go through to actual blood biomarkers, that there's a whole bunch of downstream manipulation of epigenetic results that actually determine how it is expressing itself within the human body. So it's important hinting, but we don't have the whole story in there yet. There was actually very recently a really nice a paper on MECFS that did go through this. Maybe we can leave that in the link as well, in that they took some nice transcriptomic data.
If we can, we'd like to keep in touch with both of you and talk about the pathways analysis as it develops and share any results.
Absolutely. I'd love to come back and chat about that. It's an exciting journey.
Sounds great. But in the meantime, it sounds like Mobius can be used to advance epigenetic testing. Is that right?
I think at a fundamental level, yes. That the amount of power increase we're seeing from the tests using a transformer-based engine like Mobius over previous classical techniques, XGBoost, random forest, and the like, is sufficient that I think it's really adaptable, and that we should be really keeping our eyes open for what the next set of epigenetic separations that we want to throw at the Mobius platform is.
Sounds great. So let me ask you, what's next?
Well, we're deep into pathways. That's the current mission, is figuring out what underlies and looking for partners, trying to find that new set of data. And so would love to chat with people on that. That's where we are on this particular project.
How can we follow your progress?
There are a couple of ways of following along. We have a talk at the fall MECFS conference. We'll leave a link to that one as well. There we hope to elucidate a little bit more on the pathway pieces. I think we didn't really talk about the overall parameterization of the model. That might be a little bit too detailed for some of your audience, but it's important to note that Mobius is only about an 11 million parameter model, which when you compare that to some, that's tiny. That's you compare that to the couple hundred billion parameters that are in a modern chatbot engine, for instance. We are a small, quickly executed a model that is fairly tuned towards our task, and so from an underlying perspective, it really does lead to a class of similarly trained small models for epigenetic separations that are really broad in their application.
Piyush, you had asked to finish with a few words about the size of the Mobius model.
Yeah, so I think Derek mentioned a really excellent point that our model is really quite small compared to other transformer models in the realm of transformers, right? And this means that it can be deployed, it can be scaled really easily as we get access to more data, as we want to, apply it to more conditions. And as a testament to that, our model was actually trained on just a home MacBook in 48 hours. And this means that, it can be scaled really easily and deployed. We see this model as being able to run in the cloud, in the browser, and most importantly, locally. So all of your data is private and secure. And I do want to also close off with saying that our models and our process training data sets are all open sourced and available to the public for research use. And we'll leave links down to our GitHub and our Hugging Face for that.
Derek and Piyush, thank you both for agreeing to speak about this important study. We'll check in with you again to see how your work is advancing. But until then, many thanks and good luck.
Really appreciate it. Thanks, Tom.
Thanks, Tom. Thanks for inviting us over. This was super fun for me. First podcast.